The objective of developing a low nitrogen stress screening site is to predict variety performance under low input smallholder agricultural systems that are prevalent in sub-Saharan Africa. Establishing these sites on experimental stations allows researchers to carefully manage the levels of stress and collect data in a standardized and timely manner. Researchers are also able to intervene to ensure trials generate useful data, for example, through the application of irrigation if necessary. When selecting a low nitrogen stress screening site, it is important to consider the following factors. Developing a low nitrogen stress screening site can take several seasons, as the site will need to be depleted of naturally available soil nitrogen through repeated cultivation of a crop which removes the soil nitrogen. A low nitrogen site is therefore a long-term investment and needs to be chosen with care, as it will be used for many years. Ideally, the low nitrogen stress screening site should be selected within the target population of environments for the particular breeding program, for example, mid-elevation, subtropical agroecologies. This will allow varieties to also be screened under optimal growing conditions to identify varieties that perform well under both nitrogen-stressed and high-input systems. The size of a nitrogen stress screening site will depend on the land available at the research station, bearing in mind that the site will have to be devoted to low nitrogen stress screening for many years. Typically, 1 hectare is the smallest recommended size given the investment required to develop the site. A low nitrogen stress screening site should consist of fairly uniform soils that will deplete evenly. Different soil textures within a field can result in high levels of field or environmental variation that will adversely affect trial results. A slope within the low nitrogen screening block can result in nutrient leaching and runoff to lower parts of the field, creating field variation. Likewise, the site should also not be located in a valley area where nutrients and rainwater may accumulate during the rainy season. Shading from trees and growing leguminous trees that fix atmospheric nitrogen should be avoided. Shading can affect crop growth and the soil microenvironment, leading to variation in rates of mineralization. The nitrogen stress screening site should be easily accessible and should have adequate security to ensure trials are not damaged by animals. The site should also be located where it will be possible to maintain dedicated data collection and trial management staff. Access to irrigation is important, as an expected drought can confound trial results. The site should be selected in an area where there is no heavy incidence of pest and pathogen pressure such as termites, STRIGA, or maize lethal necrosis. These can confound trial results. In this section, we describe the recommended steps that should be taken to develop a low nitrogen stress screening site, including mapping, plot demarcation, and nitrogen depletion. Once a low nitrogen stress screening site has been selected, it should be clearly demarcated and signposted as a low nitrogen site to prevent future mistakes in fertilization. The GPS coordinates of the site should be taken. A brief history and map of the site should be recorded, including notable features or past uses. This will enable researchers to understand and manage field variability. For example, This map enabled researchers to split the low nitrogen screening site at Kakamega, Kenya, based on depletion history. Researchers were also able to identify the source of waterlogging from the possession of a disused but broken water pipeline. The site should be cleared of all vegetation, free of termite mounds, and deep plowed to break any handpans and facilitate drainage. Soil testing is important to obtain the baseline soil nutrient status and to allow the depletion of soil nitrogen to be tracked over years.
A good method of soil sampling is known as the grid sampling method. The field can be marked out using a grid of 20 meters by 20 meters and samples taken at the center of each grid.
Separate soil samples should be taken at 3 soil depths: 0 to 30 centimeters, 30 to 60 centimeters, and 60 to 90 centimeters, and all samples individually analyzed. This will provide an overview of soil nutrient status at the start of site development, also including information on the variability of soil chemical and physical properties. Once a uniform site has been selected, it is important not to introduce field variability unknowingly. During experimentation, plants will remove more soil nitrogen from plots while soil nitrogen content in alleys and walkways will remain constant. As a result, it is recommended to mark out permanent plots and alleyways. This will ensure that plots are consistent season after season and do not shift into parts of the field that were previously used as alleys or borders. The fixed permanent plots also ensure that individual plot performance can be monitored season after season and that composite yield maps of the site can be generated to aid site management. Permanent plots and alleys can be referenced from a single beacon or individually using permanent metal pegs set in concrete along the perimeter of the block. Typically, a low nitrogen field plot is 5 meters long with 0.75 meter inter-row spacing. Trial yields from a low nitrogen screening site should be as representative of smallholder production as possible while enabling clear detection of genetic variation for low nitrogen performance. It has been shown that a 70% yield reduction relative to optimal conditions provides the best experimental conditions for detecting genetic differences among genotypes. A 70% yield reduction relative to optimal potential typically translates to a yield of 2.5 to 3 tons per hectare in mid-elevation environments in Africa. In order to deplete a low nitrogen screening site of residue nitrogen to achieve a 70% maize yield reduction, the site should be repeatedly sown to a single cereal variety. Maize, sorghum, or wheat can be used. Leguminous crops will fix nitrogen rather than depleting it and should not be used. Sorghum is the most effective depletion crop as it develops biomass comparable to maize but can be planted at much higher densities, up to 400,000 plants per hectare, without lodging. Sorghum is relatively drought tolerant, which makes the depletion crop easier to manage during the off-season. Sorghum can also be ratooned or cut back at flowering, allowing a second crop to develop in a single season. At the end of each depletion cycle, the entire biomass of the depletion crop should be removed from the low nitrogen screening site, including leaves, stems, and if possible roots. Depletion will be apparent when a clear border effect is visible. Typically, 2 or 3 cycles of high-density depletion are required in the mid-elevation regions of southern and eastern Africa to adequately develop a low nitrogen stress site. However, it is important to keep in mind that the higher the clay content, the longer it takes to deplete nitrogen in a field. In this section, we describe methods to characterize a low nitrogen block and monitor site depletion. Field variability is always higher under abiotic stresses. Therefore, even when the most uniform field sites are chosen, inherent field variability will still compromise the identification of nitrogen use efficient varieties. It is therefore recommended that low nitrogen screening sites should be characterized prior to experimentation to better understand natural field variation in nutrient status. This information can then be used to guide trial layout and site management. It is recommended that the site characterization methods described in this section are undertaken prior to experimentation and then repeated after every 5 cycles of experimentation. This will allow researchers to detect variation that might have been introduced through field management during experimentation, such as irrigation or fertilizer application. Due to the cost and effort involved, detailed soil sampling is not recommended for site characterization. However, the grid sampling method described in the baseline soil testing section will provide a useful overview of soil nutrient status in the low nitrogen screening block. In this example, 16 soil samples were taken using the grid sampling method at Kiboko, Kenya. The nitrate levels in parts per million in the top 30 centimeters of soil are shown in this heat map and provide an overview of nitrogen status in the topsoil. A heat map is a visual representation of data where individual values are represented by color. In this example, low levels are red while high levels are colored green to represent fertility. Generally, the nitrate level in the soil is considered deficient when it ranges between 7.5 and 15 parts per million. Soils are considered severely depleted when nitrate levels fall below 7.5 parts per million. Values around 10 parts per million will provide good experimental conditions for detecting useful genetic variation. The heat map from Kiboko suggests that the site has been adequately depleted for low nitrogen stress screening with an average nitrate content of 10 parts per million. Characterization of low nitrogen screening block by crop biomass or vigor is generally the fastest and most economical method of characterizing a site. It is assumed that crop vigor or biomass is reflective of soil nutrient status. In order to characterize a field by plot level biomass, the entire field should be sown to a single variety of cereal along the permanent rows and alleys, just as in a normal experiment. Often, it is useful to characterize a field during a depletion cycle prior to experimentation. Plot biomass can be characterized using a variety of methods such as plant height, visual estimate of plant vigor, or by measuring Normalized Differential Vegetation Index (NDVI) using a tool called the Green Seeker. All three methods are positively correlated. Normalized Difference Vegetation Index or NDVI is an estimate of green biomass and offers the most objective and high-resolution method of site characterization. A green seeker is used to measure NDVI from each plot when the crop is knee-high. It is at this stage that differences in plot vigor are clearly pronounced. Where a green seeker is unavailable, visual estimates of plot biomass using a 1 to 5 scale can be taken where 1 represents very low biomass and 5 represents the most vigorous plots. Visual scores are cheaper to collect and are typically highly correlated with vegetative index scores. However, they do not offer the resolution of a green seeker or other visual imagery tools. Individual plot scores of NDVI or visual estimates of biomass can then be used to plot heat maps using either Excel or statistical packages such as R or SAS. Heat maps can be used to help researchers plan experiments by highlighting areas of variability. Experiments should not be planted across these gradients where possible. High priority trials should be planted in the least variable area of the field. Heat maps often highlight features consistent with prior use of the site, such as old roads or trees which have been removed. In this example, a heat map of field variability has clearly identified a section of high vigor running through the center of the field. This is consistent with the position of a previous road at the field station. Rather than incorporating this highly vigorous section in experimental trials, researchers opted to set aside this area as a permanent alley to divide the low nitrogen block for easy management. The heat maps have also been used to block trials so that they are contained within areas of limited variation to provide the most uniform conditions for individual trials. Once operational, the performance of a low nitrogen stress site should be carefully monitored for the level of yield reduction and field variability. The average trial yield of the site should be determined to ensure that actual yields are close to the target yield reduction of 70% compared to well-fertilized trials. If yields are too high, then the site requires more depletion, while if yields are too low, small amounts of nitrogen fertilization may be required. Mapping trial yields and heritabilities across the site will provide researchers a good understanding of the site variation. Low nitrogen screening blocks should always be planted to at least 2 border rows on the periphery of the field to avoid the border effect. This is where plants growing on the edges of the field perform better than plants within the field due to reduced competition for light and nutrients. The site should also be maintained free of weeds both during and out of the cropping cycle, as some weeds can be leguminous and add nitrogen to the soil. In order to promote early plant vigor and maintain yield levels, limited amounts of fertilizer will need to be applied to low nitrogen stress blocks. Typically, 50 kilograms of diammonium phosphate is applied per hectare at planting, and 75 kilograms of calcium ammonium nitrate is applied as top dressing when the crop is at knee height. All other nutrients should be applied at the location-specific recommended rate. Potassium availability in nitrogen-depleted sites should be monitored closely in particular. After 4 or 5 cycles of experimentation, researchers may also consider reincorporating crop residue into the low nitrogen screening blocks to ensure soil organic content and micronutrients do not become limiting factors to crop growth. STRIGA can become a problem in low nitrogen screening blocks in regions where it is endemic. This is because maize is more susceptible to STRIGA infestation under fertility stress. Adding limited amounts of fertilizer to restore soil fertility is the most effective way to manage STRIGA. Alternatively, a volunteer crop such as sorghum may be sown in an effort to exhaust the STRIGA seed bank. The crop will then need to be removed or plowed under prior to STRIGA flowering. However, it should be noted that under severe STRIGA infestation, the STRIGA seed bank may be active for up to 20 years. Inbreds are smaller than hybrids and will therefore remove less soil nitrogen. Planting inbreds within one area of the field will therefore leave a shadow in the field of higher soil nitrogen content. To avoid this problem, an area of the field should be dedicated to inbred experiments. The development of maize varieties with higher yields under small amounts of nitrogen has the potential to significantly increase yields of smallholder farmers in sub-Saharan Africa. The key to developing and identifying new varieties is through better management of environmental variation in field trials. The objective of this video has been to highlight some experimental and management techniques that may reduce environmental variation from low nitrogen stress field trials.